In this lecture, we are going to be talking about protein extraction from mammalian cell culture. Now, protein can't be just extracted from mammalian cells grown in culture. We can actually get proteins from any number of biological materials. We can get it from tissue samples or even biological fluids if we are looking at secreted proteins. We can get it from various plant materials. We can obviously get it from cultured mammalian or plant cells or yeast or bacterial cultures as well. The basic questions we need to ask would be the same regardless of where it is that we are getting these proteins extracted from. So some of these things that we need to think about as we design our experiments are, first of all, what is it that we are going to be looking for? Sometimes we have a very new system and we don't know a lot about it, and our goal is to find out the global protein expression, how proteins are changing overall in a particular system, maybe a normal versus a particular disease phenotype, or with and without certain type of drug treatment, or it could be in response to heat or in response to a particular stimulus that we provide the cell. In that case, we are going to want to maximize the protein extraction procedure in a way that we get as many proteins out as possible, as much variety of proteins out as possible. Other times, we are looking for a particular protein of interest. In this case, maybe I'm interested in a particular protein in the death pathway, like the death receptor or caspase-3, or maybe I'm interested in a particular protein that functions in secretion of other proteins like TSAP6. Whatever that protein is that I'm interested in that case, I want to maximize its extraction. So I don't so much care about all the other proteins that are present there, but I want to make sure that every single molecule of my protein comes out, or as many of them come out of the cell as I can get out. So I would want to cater my procedure according to extraction needs for that particular protein in that case. Next, we want to think about where would I normally find this protein. So where is it that I need to get this protein out from? For example, some proteins are present inside the nucleus when they're activated, and I want to look at, if I want to look for one of their active form, that's where I want to look at. I want to extract proteins out of the nucleus. Other times, or a lot of proteins are present in the cytosol, so I don't really care about the nucleus in that case, but I want to get as many of the cytoplasmic proteins out as possible. Then other times we care about the membrane receptors. So all I care about are these proteins that are going in and out of the membrane or that are integrated into the membrane. A lot of those will have hydrophobic regions. So I would need to think about that as I'm designing my extraction procedures. Finally, there are proteins that are secreted outside the cell, where then they can affect either that cell or other cells nearby or far away. So if I'm looking for a protein that is ultimately secreted out, I can't be looking inside the cell. I have to think about where it would be going. It could be just in the extracellular matrix around it. It could be in the media, secreted into the media if it's a protein growing in culture. Or a cell growing in culture, it would secrete it outside the cell and it would end up in the media that's on top of the cells. Or it would be in one of the biological fluids if it was inside our body. So we would need to think about where it is that it's normally found or that it was expected to be found, so we know where to extract proteins out of. Next, we want to think about properties of the proteins. Does it have bindings to lipids or sugars? Does it have hydrophobic regions or hydrophilic domains? Is it charged or uncharged? Based on those, we may need to modify our buffer so that it is optimized for extraction and solubilization of that particular type of proteins. For example, if you have a highly hydrophobic proteins, you cannot have a aqueous-based lysis buffer because those proteins would not have high solubility in it and they would not be extracted the appropriate way. Next, we want to think about isoelectric points of proteins. Now, this is something that in some cases is not so important, but in other cases where we want to study maybe the activity of the protein, or we want to maintain the activity of the protein, or maybe we are interested in looking at the charges on the protein and the R groups of the protein, it is something to care about. So the isoelectric point of the protein is the point at which the net charge on the protein becomes 0. That's determined by the R groups. Each R group on the amino acid. So each amino acid is a string of amino acid as a protein, and each R group on those amino acids is going to have a particular charge depending upon what pH environment it is normally present in, or that it is present in at that given time. So at a particular pH, if you have a string of 5 amino acids, 2 of them may have a +1 charge, 1 of them may have a -1 charge, and 1 of them may be a 0, or 2 of them may be zeros. So the net charge on that protein at that pH would be a +1. However, when you move it in that pH gradient up or down, there may come a point where it will be a net charge of 0, where 2 of them are +1, 2 of them are -1, and 1 of them is 0. So the overall charge becomes 0. That's the isoelectric point of a protein. Every protein has an isoelectric point. So if we have a bunch of proteins, if we have a protein extract with, let's say, 10 proteins in there, or 5 proteins in there, or 1,000 proteins in there, whatever it is, and we run it in an electric field on a pH gradient where you have a range of pHs on a long strip, the proteins will go up and down that gradient until they reach their isoelectric point, and they will stop when they reach the 0 point. And that will be the isoelectric point, that particular pH. So it is important to know in many cases when we are isolating proteins or fractionating them, and that is something that we would need to be aware of in those cases. Finally, we want to think about, does this protein move from one place to another in response to my treatment, or in response to the stimulus I'm providing, or that the environment is giving it? Because again, that would be something important to know when you're extracting proteins. If your protein is normally present in the cytoplasm, but it gets secreted in response to the stimulus you're providing, then you would need to look in both places, or extract proteins from both places before and after treatment to show that movement. So these are some of the initial steps. Once we've kind of thought through these questions, we now want to think about the basic steps that we need to perform as far as protein purification and analysis is concerned. So obviously the very first step would be to choose the appropriate source of proteins. If I'm looking at intracellular proteins, I want to look at cells, and I want to go inside the cells, or tissue samples. If I'm looking at secreted proteins, then I want to think about biological fluids, maybe CSF, like cerebrospinal fluid, or the blood sample, or a urine sample, if it is inside the body, or in vivo. And in vitro, I would look at the media. So I want to choose the appropriate source of proteins to extract. Next, I want to think about how I'm going to be solubilizing these proteins. So I have to solubilize these proteins, extract them out of where they are, and get them into a new solution where I can look at them. Once they're solubilized, however, once they're out of their native normal environment, they are no longer going to be stable. So I have to think about what it is in my environment that is making them unstable, and how to counteract it, how to stabilize them. And we'll talk about some of those in a minute. Once we've solubilized the proteins and stabilized them, our next goal is to obviously study them. So depending upon what question we have to answer, what question it is that we were trying to answer, we would have a range of different experiments we can perform. We could be looking at physical characteristics, where all I care about is either the size or the mass, or some spectroscopic properties of the protein, or like the sequence of amino acids, for example. In those cases, it's going to be a different type of analysis that I perform, right? I may be just running them on a gel, or putting them in an NMR machine and looking at the spectrum. I may be breaking it up one amino acid at a time and sequencing it. I may be looking at any of those properties that way. I may be running crystallization studies where I try to crystallize it and look at its 3D structure. Or I may be doing it in silico, knowing the sequence of amino acids. I may be trying to predict the 3D structure of my protein. Now, on the other side, I may be actually looking at its activity, which is a little bit more involved and harder to do in many cases. So I may be looking at either its enzymatic activity, its interaction with other proteins, or its biological activity. So many times for physical characteristics outside of the 3D structure, I don't necessarily need to maintain its tertiary and quaternary properties of the protein. I can be more stringent with the type of buffers I use to extract as much of the protein out, but without keeping it, all there in its integrity, because all I care about is the size of it or the mass of it or the sequence of it. But if I'm looking at actual activity, or if I'm interested in the 3D structure, then I have to think about those things as well. I have to maintain those things. I have to maintain the enzymatic activity. I have to maintain its biological activity. So to do that, I have to maintain the active side of my protein by maintaining its 3D structure so it doesn't get denatured and doesn't get messed up. To do that, our assays have to be very specific and rapid. That's true for all of the assays, but especially important when we want to maintain 3D structures so that we can look at enzymatic activity. It is very important to choose techniques and assays that are going to be quick, that are going to be very sensitive, so that even if I have a protein that is at low abundant protein, not present in a very large quantity, I can still find it and I can still make some measurement on it. It has to be specific, so I have less chance of getting false positives or negatives. And finally, all assays, regardless of whether they are looking at physical characteristics or biological activity, should have a quantitative aspect to them. So some way for me to quantify how much was there, how much activity was there, either in absolute numbers or relative numbers at minimum, where I can compare a normal versus diseased or X versus Y. So to do that, there are some ways that we can modify our choices of extraction materials and methods. The first choice that we have is the lysis buffer, what lysis buffer we are going to be using. The basic point of the lysis buffer is to break through the cell membranes and to solubilize my proteins that I'm interested in extracting. To do that, we many times use detergents as part of the mixture. Just like detergents, Dawn cuts grease in washing dishes, similarly, using detergents in this scenario breaks down or permeabilizes the lipid membrane around the cells and allows the proteins to get out of the cell. For these buffers and for these detergents, we can have choices of ionic versus non-ionic detergents and buffers. For ionic detergents, they are going to have, like STS, they are going to have some sort of a charge, usually a negative charge. They are harshest, so you get a lot of yield of protein, but they put a negative charge on all your proteins. So now they will be disrupted as far as their structure is concerned. They will no longer be in their 3D structure. They will be long ribbons of amino acids. So if I wanted to study their activity, I can no longer do so. If I wanted to study their binding with another protein, I can no longer do so. If I wanted to look at their structure, I can no longer do so. However, they are great for looking at relative quantities of proteins, X versus Y, or protein X in normal versus diseased, or cancer versus non-cancer, with and without drug, whatever the scenario may be. They are also great for looking at just mass or size or sequence. On the other hand, if I want to worry about the structure and all that, I want to choose a non-ionic detergent. That will keep the proteins in their native form. They can be preserved, their function can be preserved in many ways, and you can study them better. You can also use a method called salting out, where we use a high ionic strength or a high concentration of salt to decrease the solubility of proteins and precipitate them out of solution. In this case, your proteins are getting precipitated out of the solution in a pellet form, and then you would resolubilize them in the buffer of your choice. This can harm their 3D structure in many cases, especially for smaller proteins. So we need to be mindful of that if you are interested in maintaining its activity. Next, we can use the pH to our advantage. Let's say that I'm interested in looking at proteins that can survive the environment of my stomach, with all the stomach acid and everything there. I can lower the pH to the environment of the stomach to pH 2 or so, and then I can look at proteins that can survive in that environment, that are present in that particular pH only, and that can still work there. I can also use this to enrich targets that I'm interested in. Again, this is one way of enriching for proteins that for the environment that I'm interested in. Another way to enrich the proteins is to fractionate my sample. I do that by taking my cell extract and kind of subfractionating it into a cytoplasmic, a membrane portion, a nuclear portion. Even mitochondrial portions can be separated out, and then I can have my secreted proteins from the outside. And this not only enriches for your target protein, but also allows you to look at movement of protein over time or with and without drug treatment, if that is something that you're interested in looking at. So it helps you localize the protein or monitor its movement over different treatments or time frames. Finally, I want to make sure that any activity modifications, any post-translational modifications that are important for its treatment are preserved. We do that by specifically looking at what glycosylations, phosphorylations, acetylations, or nitrations are present on my proteins of interest, and then preserving them by including inhibitors for destroying those processes. So the biggest problem that we have when extracting proteins, there are actually three main problems, and they all stem from the same basic problem, which is that proteins are no longer stable once they are outside their native environment. So how do we protect that? How do we preserve that? We need to know what are these things that are making them unstable. The biggest issue is that they get denatured. They have a loss of structure and activity. Every protein has a half-life, and that the protein is going to overall, over time get degraded and destroyed. Another thing is that they get proteolyzed. So they actively get destroyed by proteases that are released from the cell rupture. Normally these proteases are kind of, in their own little compartments in the lysosome or the mitochondria or wherever they need to be, and they are not working on the entire cell, destroying all the proteins randomly. However, when you rupture the cell, these proteases come out, and now they're in direct contact with all these proteins that they can proteolyze. Finally, just by being open to the air and the environment, proteins can get modified and oxidized. The phosphatase can dephosphorylate your proteins. The deglycosylation agents can deglycosylate the proteins, so they can lose their activity as well because of that. So there are multiple ways that we prevent that. The first thing is that we perform all our steps when extracting proteins at 0 to 4 degrees. Sometimes if we have a super sensitive proteins, we actually go into the cold room to do our experiments. So we physically stand in a cold room with everything at 4 degrees or below and run the entire experiment there, whether it takes 1 minute or a day. And it can be quite interesting there. Other things we can do, we can add protease inhibitors to our sample buffers so that all the proteases that are coming out are inhibited. And then we can also add phosphatase or deglycosylation inhibitors to our sample buffers as well. We do that, however, we have to keep that in mind that these inhibitors also have a half-life, and usually that half-life is relatively short. So we need to be quick in our procedures so that we can get where we need to go and run the assay as quickly as possible. So finally, we run the activity assays immediately after extraction. The longer you wait, the more activity is going to be lost, even in the most perfect environments. So our goal usually is to extract the proteins and then run set up the activity assay immediately afterwards. If you don't need to worry about the 3D structure, then you can freeze the samples and you can assay them at a different time. But if we have to keep our activity maintained, we need to run them immediately. Now, that's one part of the story, is obviously that we need to run the activity assay and then quantify it somehow. Well, to quantify it, we need to know how much protein we started with. And in general, any type of assay that we are going to look at, any type of experiment that we are going to run, we would need to know how much total protein did we get out to begin with. This is an extremely important question to ask and extremely important information to have in order to quantify our experimental results appropriately. So if you take an example of human tissue, for example, let's say that I get liver from two people, one from a normal liver and one from a person with cirrhosis, and I get 1 milligram of tissue from both of them and extract protein, I can't automatically assume that I'm going to get the same amount of protein from these two. For one had healthy liver, the other one had cirrhosis, so may had a lot of dead cells or necrosis or destruction in there. So I wouldn't have started with the same number of cells. There could also be issues with the extraction where one of them, because of the way it was with the cirrhosis, may not have had as stable proteins as well. So I cannot quantify those two, the results from those two samples without first knowing how much protein did I get out from the normal sample versus the disease sample. Similarly, when I take 2 plates of cultured cells, even when they look similar, they are not going to have the same number of cells. And the resulting protein that I extract from them is going to be different in those 2 samples. Many times, however, we have samples that are either treated in one case versus your untreated control, or that have been growing for longer or smaller period of time depending on the experimental needs. So you will be starting off with different number of cells, and hence you will get out different amount of protein. So the very first step in any experiment for data analysis purposes is to know how much protein did I start off with in each one of these samples. To do that, what we do is we run total protein quantification assays, where we just look at total amount of protein present there. We do that using many times colorimetric assays. These type of assays produce a color change that we can measure with spectrophotometry, and we can look at the absorbance that we receive from the spectrophotometric analysis to quantify the amount of protein. Some of the methods that have been around for a long time are Biuret and Lowry method. Both of these are based on binding of copper ions with the N-terminal of your peptide, and you can get a color change which is different for the Biuret versus Lowry, but in both cases it's a color change that can be quantified using spectrophotometric analysis. The issues with these 2 methods is that they take a long time to run. So typically you can take anywhere from 1.5 hours to 2.5 hours to run some samples. And on top of that, you can get a precipitate as a background noise. So your analysis may not be perfect and you may not be getting good reproducible results. A much quicker approach is to use a Kumasi blue dye binding assay. This is also called Bradford method. In this case, the dye binds to our proteins under acidic conditions, and you can change it changes the color from like a dirty brown to a blue, and you can look at the amount of blue by looking at absorbance at 595 nanometers and then quantify it that way. So when we do it this way, you can look at the protein very cleanly and you can quantify it. The issue with this one is it's a very quick and dirty approach. So because it is so quick, it can saturate very fast and it can get out of linear range. So you won't have the nice typical linear standard curve that you want in order to quantify your unknown samples appropriately. If I want to look at just the presence or absence of one single protein, then many times what we use instead is an ELISA for that specific protein with an antibody attached to it. It is something that you're also going to be testing out in one of the Labster simulations. What we typically use in our labs and also in our student classrooms is what we call the BCA protein assay. This is based on also on the copper binding to the N-terminal. However, in this particular modified assay, it is kind of an in-between between the Lowry-Biuret methods and the Kumasi Blue. So it only takes about 15-20 minutes to run, and it is a slower process, so it stays in the linear range longer. So it's not so, finicky about how quickly you need to run the spectrophotometric analysis. To do the BCA assay, we will talk about it in a second, but anytime we are running total protein and we want to quantify unknown samples, you first need to have a standard curve. So if you think back to the week 1 lab, to prepare a standard curve, we need to have a set of dilutions with known concentrations. In this case, we want to make a standard curve of a protein, so we would need to have a standard protein with known concentration, and we need to prepare a set of standard dilutions out of it. So the first thing we do in the lab for quantification of protein is to prepare a set of standard protein dilutions that we can use to create a standard curve using known amounts of water and known amounts of whatever standard protein we choose to use, and calculating their concentration based on the stock concentration of our proteins. In this case, in this example, the stock concentration of the protein is 2 milligrams per milliliter, and then we would need to make a range of concentrations using this kind of a dilution scheme. Once we have done that, we can run that standard along with our unknowns in order to prepare the standard curve and to quantify our unknown samples. So we would get a range of absorbances from our dilution range. So for the example curve, our standard curve is going to be good for up to the 2 milligrams per milliliter protein concentration, since that's the limit of the maximum. That's the maximum concentration in our known standards. The lowest would be whatever the concentration was outside of the zero for the lowest number, which in this case would be 0.25 milligrams per milliliter. So the range of the standard curve that we would prepare in this case would be from 0.25 to 2 milligrams per milliliter. Now, in order for us to most accurately quantify our unknown proteins, they need to be also within this range. And we cannot be sure of that because we don't know what concentration they are going to be, since some of them may have very few cells or some of them may have a lot of more cells, so they may give way above 2 milligrams per milliliter. So to take into account that some of your cultures may be giving you more than the 2 milligrams per milliliter concentration, we create a set of dilutions with our unknowns as well, so that we prevent a shot in the dark technique where we would need to redo the entire experiment again. And then we can back calculate to determine the amount of protein in the original sample if we had to use one of the dilutions. So once we prepare all our dilutions of our standard as well as the unknown, we take a small aliquot, usually 10 or 20 microliter from each one of our samples, and put them into a 96 well plate. So for each, sometimes we run them in duplicates. Other times, if you're just doing it as a practice in lab for classroom purposes, we just run them in single. But we pipette the exact same amount, a small aliquot from each dilution, for all our standard proteins as well as our unknown dilutions. We then add the BCA reagent to each one of the wells at the same time. Then we can shake the plates at 37 degrees for 10 to 15 minutes, and we are ready to read the absorbance at 562 nanometers for the absorbance. Once we have the absorbance, we can use those to plot our curves with the corrected values and solve for the unknowns. We're going to go into more detail about the calculations in the next video, but just to quickly recap how you would create your standard curve. You would get the absorbances from the spectrophotometer. In this case, well, 1 through 8 in each row have my standard curve numbers, so my standard dilutions, going from the highest to the lowest. Well, 8 has just the water only, so that had no protein present in there. This is what we are going to be using as a background noise. So for the remaining samples, including our unknown samples, we would subtract the value from our water only well, which in this case is well 8, and we would get our blank corrected absorbances before we can plot them into our standard curve. So we do that, and then we get our curve. Here I've shown you two different set of standards. We plot both of them, and we notice that one of them has a little bit more variability than the other. We do our trend line, a linear trend function, and we get our equation and the R square value. We can look at the R squares, and we can look at the one that is better based on the regression value being closer to 1. So in this case, row E has the more precise standard curve based on just the regression value, and that would be the one that you would want to use in order to quantify your proteins of the unknown samples. To quantify your unknown samples, we would use the equation where we would put the absorbance for the unknown samples for Y and then solve for X. This is stuff that we are going to be looking at in our assignment, and that I will be going over in the next video, where I will talk about protein analysis.